Hello and welcome to our lecture on mitosis and meiosis. As usual, I would like to briefly recapitulate what we have learned during our session on gene regulation. We have learned that every cell in our body contains cellular and functional diversity. Because our cells live in a changing environment and producing a set of genes not always required would be a waste of resources, our gene expression is highly dynamic and changes with age, after pharmacological treatment, and also during disease. So how is gene regulation achieved? The first level of gene regulation we looked at was gene regulation at transcriptional level. In order to understand this level of gene regulation, we had a closer look at eukaryotic transcription. In eukaryotic transcription, there are a few things that you need to keep in mind. You need to remember what a promoter is. Promoter is a region of the DNA that leads to initiation of transcription of the particular gene. And those promoters are located near the transcription start site of the genes, upstream on the DNA. Promoters can be about 100 up to 1000 base pairs, depending on the gene they are regulating. Within each and every single promoter, you can find a structure called Tata box, and it is an essential part of the co-promoter. The Tata box is named for its conserved DNA sequence, and this is the site where general transcription factors can bind. In addition to promoters and Tata box, many genes have one or more nearby regulatory sequences called enhancers or silencers. Enhancers are DNA sequences that influence transcription positively, silencers affect transcription negatively. And those structures act as binding sites for promoter-specific transcription factors. Again, we have two classes of transcription factors, general and promoter-specific transcription factors. General transcription factors are always required in order to facilitate transcription, whereas promoter-specific transcription factors can modulate expression level of certain genes at certain times. So general transcription factors are the most basic set of proteins, and they are always needed to activate gene transcription, and they actually modulate the binding of RNA to the DNA. In contrast, promoter-specific transcription factors control the expression of individual genes. Different promoter-specific transcription factors can be active in different cell types or developmental stage, or after pharmacological intervention, or during a disease. So those guys help general transcription factors and RNA polymerase to assemble and start the process of transcriptions. Some promoter-specific transcription factors can actually influence the transcription negatively because they bind to silencers. They can work in a variety of ways, for example, by hindering general transcription factors or RNA polymerase to access the gene. Next level of gene regulation we looked at was gene regulation at chromatin level. I hope that you remember that transcriptionally active chromatin or DNA is open, decondensed, and unactive is condensed. So we are talking about euchromatin in case of open chromatin and heterochromatin in case of the closed form. In the open form, RNA polymerase is able to bind, thereby gene expression is active. In contrast, in condensed DNA, RNA polymerase and transcription factor cannot bind, therefore there is no or very low level of transcription of the genes on the DNA. Another level of gene regulation is achieved by a process called RNA interference, and this actually acting on processed mRNA. In RNA interference, short RNA fragments bind to mRNA, generating thereby double-stranded RNA fragments. Those double-stranded fragments are recognized by certain enzymes in our cell, and this leads to selective cleavage of the RNA as a result. So, as a summary, gene regulation can be achieved by alteration of gene structure, transcriptional control, mRNA processing, by modulating stability of the RNA, and also by translational control at the ribosomes. So, what are the learning outcomes for this session? After watching all of those videos in this session, you should be able to understand the difference between mitosis and meiosis. In addition, you should be able to explain the outcomes of both of those processes in terms of number of chromosomes produced. You should know the basic steps of mitosis and meiosis, know examples of mitotic cells in the adult human body, and be aware of the evolutionary impact of meiosis, and also know how meiosis is actually contributing to development of certain genetic disorders. This is important because, well, you need to appreciate the consequences of a certain process during meiosis to understand one of our follow-up lectures on DNA mutations. Before I'm going to be talking about meiosis, we need to recapitulate a few of the basic concepts we have learned in our previous lectures. I hope that at least some of you remember that in humans we have 22 different autosomes and 2 sex chromosomes. So we have 23 pairs of chromosomes. So the full chromosomal configuration of the adult somatic cell, for example our skin cell, is 40X, because those are 23 pairs in total. Importantly, the autosomes are always nearly identical pairs. In contrast, the sex chromosomes can be different. In males, the sex chromosomes are X and Y. In females, they are 2 X chromosomes. So we refer to those individual chromosomes as sister chromatids. And the chromosomal configuration and the number of chromosomes is actually very, very characteristic for many species. For example, as I mentioned before, in humans the chromosomal configuration is 46, 22 pairs of autosomes and 2 sex chromosomes. In other organisms, this number can be very, very different. For example, in a chicken, you can find 78 chromosomes. In scorpions, just 4. Housefly has 12, and so on. But again, the number of those chromosomes is very, very characteristic for a species. Independent of the species, though, all chromosomes are composed of a pair of sister chromatids. We have a structure called centromere in the middle and kinetochores. Kinetochores are proteins attached to the centromere. And again, what you can see here are 2 sister chromatids. So this is one chromatid and this is another one. So what are kinetochores? Kinetochores are protein complexes that are assembled at each centromere. So we have the centromere region on each chromosome, we have the kinetochore, and kinetochore are attachment sites for spindle fibers that play an important role in both processes, mitosis and meiosis. They serve as attachment site for those microtubules that form the spindles. So, what are microtubules? I know that we had a look at the cytoskeleton in one of our cell biology lectures, but this is actually quite important in order to understand cell division. Microtubules are part of the cytoskeleton, and they are responsible for various kinds of movements in all eukaryotic cells. They are involved in cell division because they are forming what is called mitotic spindle. Another type of microtubules that play an important role in cell division are centrioles. Centrioles are actually organelles, and they consist of a cylindrical array of 9 microtubules, what you can see here. So, what are the main differences between mitosis and meiosis? Both of those processes are ways how cells reproduce. They are basically two ways how to achieve cell division. However, mitosis is the division of body cells. We call them somatic cells. In contrast, meiosis is a division of sex cells, also referred to as gametes. Mitosis occurs wherever more cells are needed by the body, and it always produces two new cells that are identical to each other and to the parent cell. So the genes contained in the daughter cells and also the number of chromosomes is always identical. What's quite important on this slide is also this tiny number and a letter inside of the cell. In this case, this is 2N, and this is often used to characterize chromosomal configuration. Because this adult body cell has a full set of chromosomes containing 22 pairs and 2 sex chromosomes, we call this configuration diploid, or 2N. During mitosis, one diploid cell always produces 2 diploid daughter cells. And the process of the growth and division is called the cell cycle. Meiosis, in contrast, is a very different kind of cell division. Again, a human body cell contains 46 chromosomes arranged in 23 pairs. However, our sex cells, sperms or eggs, contain only half of it. So their chromosomal configuration is 1N. During meiosis, the number of chromosomes in a cell is reduced to a half. Therefore, meiosis is sometimes also called reduction division. And this actually makes sense, because during fertilization, if a sperm meets an egg cell, this should produce a normal diploid chromosomal configuration, and this is indeed the case. This is indeed achieved by fusion of 2 cells that have 1N set of chromosomes, and we are calling them haploid cells, in contrast to diploid cells that are all other cells in our body. So the main difference is that meiosis produces haploid cells, whereas mitosis produces diploid cells, and this is something you should remember. So what cells in our body are actually subject of mitotic cell divisions? And there are actually plenty of examples. Until the 60s, it has been widely believed that many cells do not divide, and this was especially believed in case of nerve cells. However, more recent evidence is a bit more controversial. There are indeed some papers published in the last decade suggesting that cell division can take place even in the brain, suggesting that hippocampal neurons and other types of nerves could be replaced every 20 to 30 years. However, more classic cell type that is actually replaced within the body and produced via mitotic division are red blood cells. Another example are head hair cells, certainly not in my case, but most of you should have mitotic hair follicle cells. Skin cells are replaced fairly quickly. Liver cells are also proliferating cells, and so on. So, how are red blood cells produced? As some of you might know, those cells actually don't have a nucleus. However, they are produced by blood progenitor cells within the bone marrow. They divide via mitosis and then get rid of the nucleus. However, blood progenitor cells do have a normal diploid chromosomal configuration and a nucleus. The next example I showed you before are hair follicle cells. They are replaced quite quickly, and what you can see on the right hand side is actually a hair follicle placed inside of the cell culture dish in a lab, and as you can see here, those cells are happily dividing even in a dish. Another type of cells in our body that undergo mitotic cell division are fibroblasts. Fibroblasts are quite important cells if it comes to wound healing. If a wound occurs, those guys actually contribute to the wound healing quite heavily, and they do so by undergoing a series of mitotic cell divisions. They produce large number of offspring cells close to the wound and contribute thereby to the wound healing. And again, what you can see here is a picture of a dividing fibroblast in a dish. Finally, the last example of the mitotic cells I would like to show you today are mitotic liver cells. Our liver cells divide. That being said, they do not divide as quick as hair follicle cells or blood cells. However, they have a certain turnover, and this is quite necessary and important because liver cells are exposed to many different types of toxins and need to be replaced, and this also occurs via mitosis. In contrast to adult cells, also stem cells can actually divide. However, they only do so if they are activated by injury or other signals within the body. During normal cell division, something very interesting happens. If cells divide via mitosis, the telomeres at the end of chromosomes become shorter and shorter, and we call this process cellular senescence. At some point, integral parts of the chromosome are getting cut off by enzymes digesting DNA overhangs. This then affects integral genes on the chromosomes, and the cell would undergo programmed cell death. However, in tumor cells and embryonic stem cells, enzyme called telomerase is active and produces new telomeric repeats. So what you need to memorize here, that most cells are affected by cellular aging and cannot perform mitosis for unlimited number of repeats. This phenomenon is called Hayflick limit, and most of our body cells are only able to undergo around 50 rounds of division. This is not the case for cancer cells, and this is actually a challenge in tackling cancer and tumors. Again, in normal cells, after a certain number of divisions, cell division stops, the cell undergo programmed cell death and are getting eliminated from the body. However, in cancer cells with certain profile of DNA damage, this process is no longer occurring, and we have uncontrolled, unlimited cancer cell division. And this can be quite serious, as evident on an MRT performed in a brain tumor patient. On the right hand side, you can see healthy brain tissue. On the left hand, there is basically huge tumor mass which is proliferating in uncontrolled manner, and the prognosis for certain types of brain cancer is actually really bad. For example, for glioblastoma multiforme, the prognosis is a life expectancy of round about 6 months, and the patients usually don't die due to formation of metastasis, but they die simply due to the pressure which is produced by the tumor mass. So what you also could remember, this is again additional information, so it's not relevant for the exam, but stem cells are able to undergo a very interesting type of mitosis. As I mentioned before, the majority of our cells produces identical daughter cells. However, stem cells can undergo asymmetric division, where one cell is resembling the parent cell and another one produces a more differentiated or specialized offspring. But this is just additional information to give you the full picture. So, what is the cell cycle and how does it refer to mitosis? The cell cycle is actually a process that all of our cells in the body undergo if they divide, and it consists of 4 stages. And mitosis is actually the shortest one. We have certain other stages of the cell cycle where proteins are produced. We have active gene transcription. However, this is not occurring during mitosis. During mitosis, the cells are focused on producing identical offspring. And the mitosis itself can be also divided in certain phases. We have the prophase, metaphase, anaphase, and telophase. And we will gonna be looking at all of those phases in detail in our session number 2. Thank you very much.